This is my project on cell signaling for AP Bio, and to keep this video to the required 35 minutes, I'm only going to go over my chosen cell signal, but I did do the overview as specified in the directions of short and long distance communication, ligands, protein receptors, transduction pathways, and cellular response. So my chosen cell signal was epinephrine. Epinephrine is a hormone. It's also known as adrenaline. It can increase blood sugar, heart rate, contractility, which is how I understood it was how fast or how hard the heart, pumps, squeezes. And it can relax muscles that improve breathing. And the reception of epinephrine is the signaling molecule, or ligand in this case, and binds to a G protein coupled receptor in the membrane that is called a B-adrenergic receptor, sorry if I'm saying that wrong, which is part of a class of receptors called 7-transmembrane helix receptors. When the epinephrine binds to this protein receptor, it causes a change in shape in the protein receptor, which causes the G protein attached to be expelled, to leave the receptor, and it releases GDP and GTP binds to it. The receptor is a trimeric G protein, and that means it has 3 subunits, and the first subunit, is the one that actually binds the GTP and gets rid of the GDP. And the binding of GTP to the A subunit is what turns on the G protein to activate another. And this is the transduction of epinephrine. The 2 other subunits are removed from the G protein when it is activated by the binding of GTP. And the G protein along with the A subunit are what travels across the membrane to an enzyme called adenylyl cyclase. And when the G protein binds to its enzyme, it triggers the enzyme to catalyze the synthesis of cAMP from ATP. When the G protein and A subunit detach from the enzyme, it is no longer active. And the G protein and 3 subunits will eventually return to their original positions in the membrane and be ready for use again. The cAMP molecules, cyclic AMP, are the second messengers in this pathway, and the cAMP binds to protein kinase A, which is called an effector molecule in a pathway, and activates it. And activated protein kinase A goes on to phosphorylate serine or threonine, which phosphorylatase B kinase holds. And this is just a note about amplification in this pathway, which I thought was kind of important. And it says epinephrine signal can be amplified in the response stage, the first stage, when one molecule of epinephrine causes a change in multiple G proteins, and it is amplified in the production of not just one, but multiple cAMP molecules from the enzyme, which can each go out and activate their own protein kinase A. And this is the response in the cell to epinephrine. And it is the phosphorylase B kinase, which is then activated, mobilizes the storage of glycogen to break down into glucose because the anticipation of more energy needed in the body, which is what epinephrine or adrenaline signals for. And for my research, the activated protein kinase A can actually carry out a number of other functions, like activating transcription factors that would then move to the nucleus and activate certain genes, which would produce proteins and enzymes that the cell would use to produce an effect in a certain part of the body as a response to epinephrine. And protein kinase A can increase heart rate, and that's an important example, I think. And these are just some simple models that I think helped explain to me this process. And these are some models with a little more detail. This is one where the response is a transcription factor, and this is one where it doesn't specify the response, but it's just a little more detailed. That's all. Thank you for this, and I'll be turning in my sources and this slideshow as well.